Hello everyone, my name is Nour GHAZZAUI, postdoc researcher at Cribble Laboratory Limoges. I work with Dr. Deniso's team on the project entitled High Throughput Sequencing reveals that class switch recombination junctions are affected by the absence of the 3' RR cis-transcriptional enhancer. Mature B cells express on their surface immunoglobulins. These immunoglobulins are constituted of two identical light chains and two identical heavy chains. Heavy chains are encoded by the IgH locus, which is the immunoglobulin heavy chain locus, composed of a variable domain coding the antigen binding site, and a constant one containing the genes co-encoding the different immunoglobulin isotypes. In their germinal conformation, these genes coding for the immunoglobulin are non-functional. They must undergo a series of rearrangements so that the B lymphocyte can express a complete functional immunoglobulin, starting by the VDG recombination, which leads to the generation of a functional immunoglobulin repertoire, followed by somatic hypermutation, known as SHM, which occurs at the mature B cell stage and consists of pointed mutations that leads to affinity maturation toward a specific antigen, and the class switch recombination known as CSR, which occurs also at the mature B cell stage to change the immunoglobulin isotype here from an IgM toward an IgG1. So all along the B cell development, the IgH locus undergoes many breaks. A poor reconstitution of chromosomal breaks can occur, resulting in the insertion of an oncogene into the IgH locus, which is the cause of many B-cell lymphomas. This is the case of mantle cell lymphoma, where there's a translocation of cyclin D1, the translocation of BCL2 in follicular lymphoma, CMYC during Burkitt lymphoma, cyclin D1, D3, CMAF, CMYC during myeloma. All of these events are regulated by the two major cis-transcriptional enhancers of the IgH locus called EMU and the 3' regulatory region, or 3' RR. So, in the present work, we developed studies by high-throughput sequencing in order to analyze the influence of these two enhancers on the reparation of chromosomal breaks. So do they participate in the DNA reparation and therefore possibly in the insertion of the oncogene into the locus? For this, we analyzed the junctions resulting from CSR mechanism, which is a two-step process. The first one consists in the induction of DNA double-strand breaks, and the second step consists of their reparation by joining the most S donor region, which is here switch mu, and usually switch mu, and one of the downstream S acceptor region, which is here switch gamma 1, leading to an IgG1. This reparation phase is carried by the non-homologous end-joining pathway, which favors direct or blunt junctions or microhomology ones in favor of the alternative end-joining pathway, which promotes junctions by large homology. For this, and because there's a strong homology between human and mouse IgH locus, we sequenced junctions resulting from the culture of splenic purified B cells of white type 3' RR deficient and immune deficient mice models, which were cultured for 4 days with different immunoglobulin, with different cytokinic cocktail, and then analyzed by high throughput sequencing. The analysis of breaks reparation profile showed no effect on the deletion of immune enhancer on the junctions profile in all of the three isotypes, IgG1, IgG3, and Ig alpha in all of these different structures. While it's not the case in the 3' RR deletion, where there's an effect on the switch mu, switch gamma 1 junction structures with more microhomology junctions and less blunt ones. Our perspectives are to explain this difference shown in the IgG1 junction profile. It was shown that the deletion of the 3' RR affects the B cell fate with less marginal zone B cells. So we wanted to check whether this difference can be explained by different junctional signature, but depending on B cell subsets in marginal zone and follicular B cells. Our data showed no difference between marginal zone and follicular B cells reparation profiles in switch mu, switch gamma 3 junctions. We still have to analyze these profiles for both switch mu, switch gamma 1 and switch mu, switch alpha. And then if there were no difference between these two B cell subsets, we have to further investigate how the deletion of the 3' RR is affecting the structure of IgG1 junctions. We're planning to perform experiments investigating the proteins involved in the reparation pathways. It's clear that the progress to come in the treatment of mature B-cell lymphomas will come from a better understanding of the mechanisms leading to the maturation of B-cell, and particularly the 3' RR, since it participates in both breaks and their reparations. And thank you for your attention.